Hello everyone and welcome to ITLS Academy Empower the Youth. So today in this lecture we will going to study about blotting techniques, that is northern blotting and southern blotting. In this particular lecture we will going to see the working principle of northern blotting and southern blotting. We will going to see the advantages and disadvantages, and we will going to see what are their basic applications in different industries, and at last we will going to discuss some important questions. So now let's get started with the topic and let's see the introduction part. So blotting technique is the technique in which the nucleic acid or protein are immobilized onto a solid support, generally nylon or nitrocellulose membrane. Blotting of nucleic acid is the central technique for hybridization studies. Nucleic acid labeling and hybridization on membrane have formed the basis for a range of experimental techniques involving understanding of gene expression, organization, etc. So what is a blotting technique? So blotting technique is a technique in which nucleic acid or protein, they are immobilized into a solid support, and solid support, it is generally of a nylon or nitrocellulose membrane. Means the solid support is generally of nylon, or sometimes it is nitrocellulose membrane. So the blotting of nucleic acid, it is a very central technique for hybridization studies. So whenever you want to study any hybridization, so blotting of nucleic acid, it is a central technique for this study. Then this nucleic acid labeling and hybridization on membrane have formed the basis for the range of experimental techniques that involves the gene expression, organization, etc. So this is what a blotting technique is. So generally these 2 points are you should always remember that what is a blotting technique, which type of solid support is there, and in which studies it is most commonly used, or it has a great application in which studies. Now moving to the next slide. Then identifying and measuring a specific protein in a complex biological mixture such as blood have a long been important goal in scientific and diagnostic practices. So whenever you want to identify or you want to measure any specific protein in a complex biological mixture, like for example blood, so they have been an important goal in the scientific as well as diagnostic practices. So whenever you want to have to achieve a goal in the scientific, or whenever you want to have any diagnostic process practices, so this identifying and measuring of a specific protein in a biological complexes such as blood, so this is an important in these 2 practices, that is scientific as well as the diagnostic practices. Now comes more recently the identification of abnormal genes in genomic DNA has been increasingly important in clinical research and genetic counseling. So this more recently we all are aware of this term, that is abnormal gene. So identification of such an abnormal genes in the genomic DNA, so they are becoming and increasing important in clinical research and genetic counseling. So there also this blotting technique is used whenever you want to identify the abnormal genes in any genomic DNA. So you can simply say that it has a great application or importance in clinical research and genetic counseling. Why? Because it is used to identify the abnormal genes that is basically present in the genomic DNA. So the blotting techniques are also used to identify unique protein and nucleic acid sequences. So whenever you want to identify any unique protein, okay, whenever you want to identify the nucleic acid sequences, so this blotting techniques has a great advantages, or is generally used to identify the unique proteins and nucleic acid sequences. They have been developed to a highly specific and sensitive and have become an important tool in both molecular biology and clinical research. So it has been developed to be a very highly specific, and they are very sensitive, and they have become an important tools in both the molecular biology as well as the clinical research. So in this point we have already discussed about the clinical research, that at whenever you want to identify any abnormal gene, okay, in a genomic DNA, so you use the blotting technique. So it has a great it become an important tool for the clinical research as well as in the for the molecular biology also. Now comes the general principle. So what is the general principle behind this blotting techniques? So the blotting method are fairly simple and usually consist of 4 steps. Okay, so the blotting technique is a very simple, means it does not require any rocket sciences, or it does not require any complex steps. So it is very simple, and it generally consists of the 4 separate steps. The first is the electrophoretic separation of protein in or nucleic acid fragments in the sample. Second is the transfer to the immobilization on a paper support. Third is the binding of analytical probe to target molecule on the paper. And fourth is your visualization of bond probe. Okay, so I'm just marking every thing with a different ink. So the first step is the electrophoretic separation of protein. Okay, second step is the the first is till protein and nucleic acid fragment in the sample. So this one is your first step. Now when it comes to the second, so it is to the transfer and immobilization on the paper support. This is the second step. Then the third step is the binding of analytical probe to a target molecule on the paper. This is the third. And the fourth one is your visualization of the bond probe. So generally it has only these 4 steps. Okay, so now let's quickly understand this thing, that is the molecule in a sample are first separated by electrophoresis. So the first step generally required is the separation of molecule in the sample. So molecule in the samples, how they get separated? By electrophoresis. Okay, then they are transferred to an easily handled support medium or membrane. Then after that they are generally transferred to the support medium or membrane. That is the second step. Then this immobilization and of the protein or DNA fragments, it provide a very faithful replica of the original separation. Replica means a true you can say a carbon copy of this original separation. So immobilization, what does this immobilization do? The immobilization and the protein or DNA fragment, so this provide a faithful replication of the original separation. Means what is separated, so it provide a faithful replication, and it facilitate the subsequent biochemical analysis. After being transferred to the support medium, the immobilized protein or nucleic acid fragment, it is localized by using probes such as antibodies or DNA that specifically bind to the molecule of interest. Okay, so after that they are being transferred to the support medium, the immobilized protein or nucleic acid fragment, which is localized by using a probe like for example antibodies or DNA, thus specifically bind to the molecule of interest. Then what they do, they are binded to the molecule of interest. then comes the last step, that is finally the position of the probe that to be bound that is bound to the immobilized target molecule is visualized using a auto radiography. So three main blotting techniques have been developed. They are known as southern blotting, northern and western blotting. So basically, blotting technique, if I say blotting technique is divided into three parts, that is northern blotting, southern blotting and western blotting. One addition is the southern west blotting, that is an additional, but the main you can say the blotting technique is northern, southern and western blotting. Okay, so we in this particular lecture we will going to complete the two, that is a northern blotting and the southern blotting. After that, in the next lecture we will going to cover western blotting and southern western blotting in the next lecture. So now let us understand what is a northern blotting. So northern blotting is the method used to detect a specific RNA sequence in a sample of mixed RNAs. So what is a northern blotting? So northern blotting is a method or a blotting technique that is basically used to detect RNA sequences in a sample of mixed RNA. So whenever there is a whenever you can say whenever there is a mixed you can say RNA, it is a for example this is a mixture of mixed RNA. Okay, so whenever you want to identify any specific RNA, this is the mixture, but whenever you want to identify, let's say this specific RNA, then which type of technique is used? Northern blotting technique is used. So again it has the following steps in the northern blot. So again there would be the three steps that is there in the northern blotting. So the first step is electrophoresis. Electrophoresis again it segregate RNA samples based on their size into different bands. So what is the first step? They get means it segregates the RNA samples based on their size into different bands. Now the second one is the transfer, means the RNA band in the gel are moved to the membrane by the capillary action. So the second step says that RNA band in that is present in the gel, they are further moved to the membrane by the capillary action. Then the third step says identification of specific sequence. Whenever you want to identify any specific sequences, so the target RNA sequence, they are identified by hybridization with a tagged oligonucleotide probe made up of DNA. So the northern blotting can be utilized in gene expression research because it can identify the specific RNA sequences in the sample. It can also assist the diagnosis of various type of diseases. So as we have understood that the RNA band in the gel, they are moved to the membrane by the capillary action. Then the target RNA sequence, means what is your target, means which RNA you want to detect, means that the target RNA sequence, they are further identified by hybridization with a tag of the oligonucleotide probe. Means you use the probe that is a oligonucleotide probe that is made up of DNA. With the help of this you identify the RNA sequence by a process known as hybridization. But here which type of what is the probe that it is using? Oligonucleotide probe made up of DNA. So the northern blotting they can be utilized in gene expression research. So how in fact in interviews also this question is generally there, that Why northern blotting is utilized or used in the gene expression research? So because it can identify specific RNA sequence in a sample, and it can also assist in the diagnostic of various type of diseases. So this is the reason. So this is again an important question from the interview point of view, or whenever you are preparing for an examination. Now let's move to the steps. So first step is to extract RNA from the sample. So this step is here, electrophoresis is done. So you want to extract RNA from sample, so separate RNA by size. So according to their size, RNA is separated using denaturing gel electrophoresis. So which type of electrophoresis here it is using to separate the RNA sample? Denaturing gel electrophoresis. Okay, now the third step is they are transfer and fix these separated RNA molecules from the gel to a nylon membrane. So they are further transfer and fix the separate RNA molecule from the gel to the nylon membrane. Then further it moves to the probe for RNA of interest using a nucleotide probe. So here you are using a they are using a nucleotide probe for RNA of interest. Like which type of probe? This oligonucleotide probe that is made up of DNA. Then the last step says that they generally visualize the document bands using a digital imaging system such as NUES or Sapphire FL biomolecular imaging system. It can carry out the phosphor imaging for use with the radio labeled probe. So these are the 4 steps that are generally there in the northern blotting. So this is how the northern blotting take place. This is the mixture of RNA. You want to extract your interested RNA, or you can say interest of RNA you want to extract from this. So with the help of electrophoresis tank, you use this for the separation. So this is the whole setup of how the northern blotting take place. Okay, so you can, if you want, you can take a screenshot of this. Now comes the southern blotting. Again, southern blotting is the next blotting technique, and this method is again used for detecting of a specific DNA sequence. So here if you see that northern blotting and southern blotting. Okay, so northern blotting detect RNA, whereas southern blotting detect DNA of interest. Okay, so this is the basic difference between the two. Okay, so the southern blotting it is a method of detecting specific DNA sequences in a sample of the mixed DNA. So what are the 4 steps? Again, the 4 steps are same. That is, first is electrophoresis, second is the transfer, and the last is identification of specific sequences. So if we talk about the first step, then the first step says that electrophoresis, so it segregates the DNA sample into the separate band based on their size by a gel electrophoresis. Okay, so here also the DNA gets segregated into a separate band, and on the basis of their size by the gel electrophoresis. So here which type of electrophoresis is used? The gel electrophoresis. Then the transfer of the DNA bands are means, then the second step says the transfer. So the DNA bands are transferred by a capillary action onto the nitrocellulose membrane, or you can say nylon, in a contact with the gel during the transfer process. Now comes the third step, that is identification of specific sequences. Then the particular tagged oligonucleotide sequences known as hybridization probe, that is 100 to 500 bp, is hydrolyzed with the membrane tagged sequences. Strategy has an impact on hybridization, and they are influenced by the temperature and salt content of hybridization buffer. Low strategy is defined as low temperature and high salt concentration, reducing the specificity of the hybridization, while high stringency is defined as a high temperature and low salt concentration which increases the specificity. So again, this term is very important. And stringency is defined as an impact of hybridization. So and it is basically influenced by two factors, that is temperature and salt content, okay, of hybridization buffer. So whenever there is a low stringency, so it is defined as a low temperature and high salt concentration. So the temperature is low but the salt concentration is high. So in this case, when the temperature is low and salt concentration is high, then the specificity of hybridization, it get reduced, okay. But while when the high stringency is defined as temperature is high but the salt concentration is low, means vice versa, okay, so it increases the specificity. So this is about the southern blotting. Now let's understand the steps that are involved in southern blotting. So these are certain steps that is same. Let us understand. So they extract DNA from the sample, then the fragment of DNA with the restriction enzyme. So you use the restriction enzyme to cut the fragment of DNA. Then you separate DNA on the basis of their size by using a gel electrophoresis. Which type of electrophoresis is using here? Gel electrophoresis. Then you transfer the fixed separate DNA molecule from the gel onto the nylon membrane. Then the fourth step says you transfer the fixed separated DNA molecule from the gel into the nylon membrane. Then using a probe for the DNA of interest, then probe for the DNA of interest using a nucleotide probe. So which type of probe you are using? The nucleotide probe. And then the radio labeled probe, they can be analyzed after they are detected using a X-ray film or a phosphoral imaging using a laser scanner. Example such as New Azure Sapphire, that is same that with the help of this only we are detecting the RNA also. Then same device is used for the extraction of DNA or identification of DNA. So this is a genomic DNA. With the help of a restriction enzyme, that is endonucleases, you cut this DNA fragment and you put it into the agarose gel electrophoresis. It has have a negative and positively charged, and because of the high molecular weight and low molecular weight. So in the negative charges, the upper is a high molecular weight, the low then positive side they have a low molecular weight. Then you put it into the blotting buffer. They have a agarose gel, and the plate is you can say nitrocellulose or nylon fiber. Then they have a probe, that is a nitrocellulose or nylon filter. In this step that we have studied in the step 5. Then they are exposed to the X-ray film, and here you get your gene your interest DNA, okay. The fragment that you wanted to separate, you get this when in the X-ray film. So this is the whole setup of the southern blotting technique. As far as the two blotting is concerned, both are somewhat same, steps are same, only here you are separating the specific DNA sequence and here you are separating the specific DNA sequences. Now let us understand what is the basic differences between the two. This question is there 110% in examination, so you must be aware of. Okay, so now let's quickly understand the differences between the two. So the first difference is southern blotting and northern blotting. Okay, separation of DNA, here the separation of RNA. Denaturization, say it needed denaturization, but in northern blotting denaturization is not needed. Then membrane, which type of membrane it is using? Nitrocellulose filter membrane, but here they are using amino benzoyl oxymethyl filter paper. Then comes the hybridization. So hybridization, which type of hybridization? DNA and DNA, but here RNA DNA hybridization is there. So all these differences you should be known. You can just simply take a screenshot of these differences. Okay, so this is the difference between the northern and southern blotting. Now moving to the application. So if we see the application, then a standard for the study of gene expression at the level of mRNA, and then the second is they have a great application of mRNA transcript sizes. Whenever you want to study RNA degradation, you can refer to this technique, that is northern blotting techniques. Then whenever you want to study DNA RNA splicing, then this technique is used. This technique is also used to study RNA half life, and they are often used to confirm and check the transgenic and knockout mice, means animal. So these are certain applications of northern and southern blotting, or in simple words of the blotting technique. So you must be aware of the application. So that brings to the end of the lecture. Let's quickly understand and see what all we have covered so far. After that we will going to discuss some important frequently asked question. So the first thing that we have covered is the blotting technique. We have seen that what is a blotting technique, and then we have seen the types of blotting technique. Okay, then we have seen the general principle, means what are the on which principle blotting technique is based on, what are the steps that are used in the blotting techniques, and further we have seen what is the northern blotting, what are the steps that are involved in the northern blotting. Then we have seen the steps with the help of a flow diagram. This is a whole setup of the northern blot. Similar cases with the southern blotting. We have seen what is southern blotting. We have seen the steps that are involved in the southern blotting. Then we have seen the steps that are involved in the southern blotting. We have seen the whole setup, how the southern blotting take place. Then we have seen the most important slide, that is the difference between the northern blotting and southern blotting. Then at last we have seen the application. Now let's quickly understand what are some important questions that can be asked in the interview or sometimes in your examination. So which enzyme is most often used in the blotting procedure? So the answer for that is horseradish peroxidase or phosphatase labeled secondary antibodies are most commonly used enzyme to labeled secondary antibodies because they produce a colorful product when employed with the substrate. So this is the answer for this question. Then comes which gel is used in the northern blotting, means which type of gel. So the northern blotting encompasses denaturing formaldehyde agarose gel. So agarose gel is used, that is a formaldehyde agarose gel is used as a gel in the northern blotting. Then comes what is the main purpose of blotting, means what is the main principle or aim behind the blotting. So blotting is a common diagnostic technique that is generally used in the molecular biology. Why? Just to identify protein and nucleic acid. So this method immobilizes the target molecule on the support such as nylon or nitrocellulose membrane. So these are certain important questions that can be asked in the interview. So now that brings to the end of the lecture. I hope you have understood the lecture. If you have any doubt, any queries related to the lecture, you can simply leave your queries in the comment section below. In the next lecture, I will be answering all your queries. This is our helpline number, students, that is 7080833450. You can simply call on this helpline number and get updated for any admission or in any of the respective courses you want to take. Or you can simply visit our website, that is www.idlsacademy.com. Thank you for watching. Please do like, share and subscribe. And yes, students, don't forget to press the bell icon for the latest update. Thank you.